if agricultural waste could help plants grow faster and cleaner.
Welcome to our study of carbon quantum dots derived from honey banana leaves, a sustainable solution for duckweed cultivation.
Banana leaves, a common agricultural waste, may hold the key to sustainable plant growth. In this study, derivation of carbon quantum dots from honey banana leaves or Musa x Parasidiaca L for duckweed growth, we explore how nanotechnology and nature can work together for green innovation.
This research focuses on turning waste into wonder, using banana leaves to create carbon quantum dots or QCDs that enhance the growth of duckweeds, a fast-growing aquatic plant with diverse applications.
Duckweed isn't just a simple pond plant, it's a powerhouse of nutrients. It contains proteins, carbohydrates, and fats, making it useful for foods, pharmaceuticals, and bioremediation. However, its full potential relies on how effectively we can cultivate it.
In reality, studies such as of that funded at 2023 reveals that duckweeds have long been used in traditional medicine, cited in 8 ancient texts, for treating 24 human diseases including skin disorders, abscesses, facial issues, and even hair loss.
Yet, despite their benefits, duckweeds face challenges in survival. While they can double their biomass every 24 to 48 hours under ideal conditions, they compete with algae for nutrients in the same habitat.
That's why it's vital to develop a growth enhancer, a fertilizer that helps duckweed thrive despite these challenges.
Because these so-called weeds might actually be nature's hidden heroes, capable of benefiting both society and the environment.
That's where carbon quantum dots come in. Nano-sized fluorescent particles can improve plant growth and photosynthesis. Our study aims to derive these QCDs from honey banana leaves, an eco-friendly and renewable resources abundant here in the Philippines.
This approach supports sustainability, waste reduction, and green nanotechnology, using waste to create growth. That's the power of green nanotechnology.
The primary goal of our study is to synthesize and utilize carbon quantum dots derived from honey banana leaves, is to explore their effects on the growth and development of duckweed. Establish a low-cost, eco-friendly method of producing carbon quantum dots through green synthesis, free from toxic chemicals.
Evaluate how varying CQD concentrations affect duckweed's growth, chlorophyll content, and vitality.
Identify the optimal concentration that boosts plant performance without toxicity.
And finally, characterize the CQDs using analytical and spectroscopic methods to confirm the optical and chemical properties.
Ultimately, this research envisions a sustainable model that unites nanotechnology, plant biology, and environmental science.
Proving that derivation and ecology can grow together.
This study followed the randomized complete block design or RCBD consisting of 3 groups. The experimental group treated with CQDs, the positive control group exposed to sunlight only, and the negative control group with no treatment and no sunlight. After treatment, all groups' performance was assessed by counting the number of duckweeds in each container, allowing us to compare the growth efficiency of the CQD treated samples.
We followed standard procedures for producing and testing carbon quantum dots to ensure reliability and comparability with previous studies.
However, our methodology was extended and adapted to improve existing studies and match the specific properties of our CQDs.
We collected 3 kilograms of honey banana leaves from Barangay Carriedo, Cayuga, Pangasinan. Only healthy leaves, without black spots, dried edges, or yellowing versilite. The leaves were washed with sterilized water and cut into small pieces for processing. Extraction was conducted at Virgin Milagrosa University Foundation using the one-step hydrothermal method. The chopped banana leaves were dissolved in 100 ml of water per kilogram in round bottom flasks.
The solution was heated in a Teflon line autoclave at 120 to 220 degrees Celsius for several hours. After cooling, the resulting product was washed with the ionized water to remove byproducts, yielding the carbon quantum dots.
To stabilize the CQDs, we prepared chitosan from clean and ground crustacean shells. The powdered chitosan was then added to the CQD product at a 1:1 ratio, ensuring stability and even dispersion.
9 sterilized containers were prepared, each with 5 duckweeds. Replication 1 received 5 ml of CQD. Replication 2 received 10 ml. And replication 3 received 20 ml.
These were compared with the sunlight-only positive controls and the untreated negative controls. These containers were left undisturbed for observation.
After the experimental period, the number of duckweeds in each group was counted and recorded for analysis. The data was compared across treatments to determine the CQD's effectiveness in promoting duckweed growth.
Our results showed a clear pattern. Duckweeds treated with CQDs grew significantly faster and healthier than the control groups.
First experiment, the CQD-treated group grew 14 duckweeds, compared to 12 for the sunlight-only group, and none for the untreated group. In the second set, 17, 11, 5 respectively, and in the third, 19, 13, and 5 respectively.
These results confirms that carbon quantum dots from banana leaves can enhance photosynthesis, nutrient absorption, and overall plant vitality.
Our study proves that carbon quantum dots derived from honey banana leaves can significantly promote duckweed growth in an eco-friendly way.
It supports green nanotechnology, waste utilization, and aligns with the Sustainable Development Goals, promoting responsible production, innovation, and climate action.
This research also highlights how science can transform agricultural waste into tools for sustainable development and environmental improvement.
From waste to growth, from leaves to life.